we've injected all the chemo here. So the next thing I do is I'm just gonna inject some saline to push out. There's gonna be dead space in the actual Renovo catheter itself. So basically to push whatever remaining chemo is in the catheter into the patient. I'm injecting about 5 mLs here. Just clear out any chemo. All right, so now we've pushed the chemo in, and now we're going to deflate the 2 balloons here. I always find that it takes a little while for the balloons to come down. We're using about 1/3 contrast in here. Maybe if we used a little bit less, it would come down a little bit faster, but it's not a big deal. You can see the proximal balloon coming down faster than the distal balloon. You can see that the proximal balloon is all the way down. Distal balloon, we had put more contrast in it, and at the I think I put a little 100% contrast at the end, so it's just taking a little while longer to come down, but not the end of the world. It'll come. Although I've never encountered this, but in a situation where the balloon wouldn't come down at all, if you just pull it back to the sheath, you'll end up rupturing the balloon. So I haven't had to do that, but if you had to do that, reasonable troubleshooting maneuver. Coming down slowly. But basically what we're going to do is after the balloons come down, we're going to pull out the Renovo catheter, do another run to make sure we haven't caused any injury, and then call it a day. It's coming down just slowly. When we first started doing these procedures, the hardest part about it was, there were some things that we just learned from each other and from other operators when we started doing these procedures, like using a steerable sheath and the certain wires and how to get things in place. Yeah, there's always some nuances. The other big thing about it is the logistics of the procedure is how much chemo do you need? Who's going to bring you the chemo? Is the chemo going to be here on time? Learning how to use the injector for the chemo. It sounds like a really basic thing, but not everybody here knows how to set it at those parameters. Those were those were the learning things. Now that we kind of have all that stuff in place, it really doesn't take that long. It's getting in there, doing an angiogram. A lot of times, for me, the biggest limiting factor is how long it takes us to get our ACT at the level that we want to be. But that's a good procedure. It takes less than an hour and a half. Just the important things that we've learned along the way is, like I mentioned, the steerable sheath to me is extremely, extremely valuable. Okay, the balloon has gone down yet. Obviously, make sure the patient is fully anticoagulated. Sometimes when you have, really with the SMA, you have a little branch that comes up. You sometimes have to really adjust the balloons, as I mentioned, like we did in this case, and where the proximal balloon is almost outside of the SMA. And sometimes if you blow up the balloon a little bit more, what it'll do is it might have already been occlusive, but it might just expand a little bit more to cover one of those branches. And that's really the key. But outside of that, the thing about this procedure is it can be done by any, skilled interventionalist. Okay, so our balloons are down. So next thing we're going to do is we're going to go ahead and pull out the Renovo catheter. I'm going to leave the wire in place just in case if there's any injury. See that it's coming out very nicely, smoothly here. Wire? Okay. Good. Okay, so we've taken it out. Next thing we're going to do is we're going to do a run. Do you got some contrast? And can we get another ACT syringe, please? Oh, is it? Can I use this? Yes? Okay. All right, we're going to start with the run here. You ready? I want you to hold your breath right now. Don't breathe, don't move. Beautiful, beautiful. All right. So you can see that here the SMA looks very good here. I don't see any evidence of injury or any dissection. Those balloons, what I like, I was, before we started doing these procedures, I was a little bit concerned about shutting down the SMA for 20 minutes. But we have not seen any occlusion, and those balloons are pretty soft. They're compliant balloons, and they're not causing any injury here. So I feel pretty good about this. So next thing we're gonna do is we're gonna go ahead and pull this thing out. We're gonna close the groin. I'm gonna kind of. We treated a decent number of patients as a part of the TIGER-PaC clinical trial, and but my experiences on the patients that we've treated is, number one, the procedure is very well tolerated. We do the procedure with moderate sedation. Most of the patients feel very little. Sometimes they do get a little nauseous, and we give them Zofran. Again, most of these, or not most of them, all of them have been with moderate sedation. Number two, we discharge the patients the same day. Yes, we heparinize them. I tend to close the groin. They go home the same day, several hours after the procedure. I tend to see all the patients for follow-up in 1 week. And usually a lot of them will experience a little tiredness, the first day or two. But when I see them in 1 week, they're usually back to their baseline. And that really correlates with some of the pharmacokinetics that we're seeing in the clinical trial. As we're putting the chemo directly between these two balloons, we're getting high concentrations of chemo to the tumor, but you're not getting a lot of systemic chemotherapy, and that has been already shown in studies, and therefore they don't have the same toxicity compared to systemic therapy. And with this patient, we specifically asked him, which one feels better, and he said, it definitely has less side effects with intra-arterial administration. That's, what has been seen in the studies as well. And again, at least before the trial, we treat the patients every 2 weeks, so we haven't really seen any issues with that whatsoever. Again, some of the important things, as we said here, is we are fully anticoagulating the patients, and we do put the patients on antiplatelets to further decrease the risk of any vessel thrombosis from occurring.